we classify things by a handful of small surface markers. Just to bring this back for a moment to kind of chromatin accessibility states, do we think that when folks are working on therapies like an iPSC-derived immune cell, classifying this as, okay, it's a CD8 positive T cell because it has CD8 on the surface. I guess technically that's kind of tautologically true, but it's something that I think if we were to take a look at that CD8 positive T cell versus another CD8 positive T cell versus a third one, obviously very, very different cell states. And I think as folks now with, RNA-seq and other types of technologies to better understand kind of genome-wide expression levels, and also things like ATAC-seq to understand genome-wide chromatin accessibility states, can really start to understand the complexities of the different subpopulations that we're talking about here and the differences within them.
Hello and welcome to Precision Signals. I'm Khozin. My guest today is Dan McHugh, investor at Yosemite, a fund that sits at the convergence of deep science, venture capital, and mission-driven philanthropy. This is a story about a decade-long experiment in how we fund innovation. For the past 10 years, Dan has worked alongside Reed Jobs to build a unique sandbox, initially inside Emerson Collective, the family office led by Laurene Powell Jobs. Reed joined Emerson in 2015 to lead their oncology efforts. He and Dan, friends since their days fencing together at Stanford, partnered to dismantle the traditional walls between charity and private capital. They built a tool-agnostic strategy where no strings-attached philanthropic grants are used to de-risk high-risk, high-reward research. And venture capital is then deployed to scale successful discoveries into companies. Yosemite is the evolution of that partnership. It's an independent entity created to bring this dual-engine model to a global scale with a single audacious mission: to make cancer non-lethal in our lifetime. In this conversation, Dan takes us inside Yosemite's portfolio. He describes a Cambrian explosion of new therapeutic modalities that are moving us beyond traditional approaches. We discuss their bets on antibody-drug conjugates, radiopharmaceuticals, and neoantigen vaccines. We also explore the frontier of induced proximity, going beyond simple protein degradation to engineer entirely new cellular behaviors. Dan's personal journey is equally intriguing. He began at the bench in the Greenleaf Lab at Stanford, ground zero for modern chromatin biology, and went on to become the co-founder and CEO of Tune Therapeutics, a company focused on epigenetic editing. At its core, this is a conversation about incentives. We discuss how Dan and Reed are bridging the valley of death in drug development, create a system where good ideas, whether from a Nobel laureate or first-time PI discovering RNA on the surface of the cell, are never allowed to fall through the cracks. Let's step into the conversation and trace the signal beneath the noise. Hi, Dan. Welcome to Precision Signals.
Hey, Sean. Great to see you.
Excellent. Dan, you've occupied such an interesting coordinate in our industry, sitting at the intersection of very deep technical knowledge and scientific know-how, venture capital, and mission-driven philanthropy. So I've been really looking forward to this conversation.
Awesome. Thanks. Yeah, super excited to be here and talk about some of this stuff with you.
So before we go into your professional experience, I want to start with Dan, the person, if that's okay.
Sure.
All right. Excellent. So Dan, can you tell us where you grew up and what drove you to science and specifically cancer research?
Yeah, sure. Happy to. I think I was fortunate to grow up in the biotech capital of the world, Boston, or really, more accurately, Belmont, Massachusetts. So it's a suburb just right outside of Cambridge. What drew me to science, I was thinking about this question a little bit this morning. I think looking back, it originally was actually a project back in high school. Our first chemistry class, we were all assigned a different type of technology, and I got recombinant DNA before I knew what that was. And I think at that time, just really seeing kind of, frankly, fish glow was a pretty cool experience. And it wasn't really at that time thinking through the potential health implications of this whole field, and not any deep industry experience despite growing up in Boston with my family in the space or anything like that. But it was really, wow, you can do really cool stuff with DNA and genetic engineering, and folks can already make fish glow. I didn't realize that was a thing people could do. And then I think from there, I was fortunate enough that my high school was a part of Boston University, so got exposure to a lot of different speakers that were coming through and stuff like that. And I think through a combination of got a chance to work in a proteomics lab, which was a cool experience, and first really the taste of kind of that lab bench science work, but also importantly, Jay Kiesling, who's a professor at Berkeley in the synthetic biospace,
Hmm.
came to BU to give a lecture that I attended, and you know, at that time, he was presenting on his work around re-engineered yeast to produce an artemisinin precursor through fermentation. And, I think that was really the first insight I had into, wow, this has the potential not just to be cool, but, this sort of technology can have a massive impact business-wise, patient-wise, something that has the potential to really dramatically change the cogs of a therapy like that for that sort of indication. I think, the implications were immediately clear. Not sure that exact, story has evolved that way in terms of impact, but I think still like an amazing example of the potential power of something like synthetic bio in terms of translation to human health. So from there, I got really interested in that kind of translational element of academic biology science and into patient impact and exploring different career choices, realized that venture capital really was one that you got to try a bunch of different things instead of just working
Hmm.
on one project at once. And frankly, that was the thing that attracted me to it the most. So ever since kind of middle of high school or so, I've wanted to do early stage biotech investing, essentially
Oh, wow.
get involved in a bunch of cool early stage projects.
That's very interesting. So this goes back all the way to your high school experience,
Yeah.
which is remarkable, Dan. So you were one of those people who actually listened to things in high school. That's fantastic. Despite the fact that you were interested in finance, let's
Yeah.
say, going all the way back to high school, you started your undergrad. When you were undergrad, you were quite academic, and you were at the Greenleaf Lab,
Yeah.
if I'm not mistaken, which, for those that don't know, is responsible for a lot of what we know today about the chromatin, the TAC-seq emerged out of that lab, in addition to a lot of other innovations around epigenetics. So, what drew you to that theme, and how was your experience there?
Yeah, totally. And actually, we need to go back a little bit further before the Greenleaf Lab in terms of this story because as I mentioned in high school, worked in a proteomics lab, so was somewhat
Yeah.
interested on the tool side already, but which is a space that the Greenleaf Lab continues to innovate in. But actually, my first lab that I worked in at Stanford was with Han Li Ji at the Stanford Genome Technology Center around studying genetics of Lynch syndrome patients, and it was actually in that lab that I got to know Reed better. We've been on the fencing team together, but that was our first time actually really working together back in 2011. But frankly, I think just got very serendipitously, fortunately, assigned to a really awesome lab tech, essentially as his like undergrad buddy to help do some of the work. And that was Jason Buenrostro, who at the time was a lab tech, it was before starting his grad work at Stanford, which he began the next year. And when he was starting his work in the genetics department, Will Greenleaf's lab was fairly new, and they've been talking about a number of projects, and he ended up joining that lab for his PhD. And I just viewed it as a good opportunity to get a slightly different research experience, but still continue to work with him in a space I was interested in. So a lot of credit, frankly, to Jason and the work that he was doing and dragging me along with him to Will's lab to continue some of that. And I think it was a really awesome experience. It's a lab that, continues to innovate in a number of different areas. But as you said, kind of chromatin structural bio
Mhm.
work and the tools that enable the study of that are, a critical part of what the labs worked on and what Jason worked on during that period with the development of a TAC-seq that, I'm sure many listeners are familiar with. We see it in our portfolio companies and grantee researchers all the time as a fairly common assay to understand the chromatin accessibility landscape of cells. So, really cool to get to work with him and Will as that was being developed, and I think that definitely has sparked a I guess more than a decade and a half long interest in chromatin accessibility and
Sure.
epigenetics and the effect of that on different expression levels and disease pathology, which is filtered through to some investments that we've gotten involved with that I'm really excited.
Right. Yeah, definitely looking forward to getting into those investment decisions. So it seems like when you were at Stanford, a lot of basic science research was about cataloging things, let's say,
Yeah,
even translational and clinical. I always joke about the fact that in my specialty, thoracic oncology, the way we classify um, lung cancer is very descriptive and linguistic. We
yeah,
look at the biospecimen under a microscope. We categorize the cells as either being small cell, and but we don't say large because the other category is quite diverse of cells of varying sizes. So we literally made up a phrase, non-small, which miraculously got codified in the 1960s by the WHO as a histological classification. So very descriptive, and that's a legacy that in some cases even continues to this day. But it seems like you may have had an engineering mind very early on, where uh, descriptive research wasn't enough. You wanted to understand and then manipulate the biological ecosystems, it seems.
yeah. No, I absolutely appreciate it, and that's the non-small cell lung cancer is a really funny example, I think, on that kind of historical categorization,
Right.
what we get stuck into, and I think that's true across wide areas of different biology, right? I mean, if you even think about it outside of the kind of disease classification side, but also on the individual cell side, I mean, we classify things by handful of small surface markers. Just to bring this back for a moment to kind of chromatin accessibility states. Do we think that when folks are working on therapies like an iPSC derived immune cell, classifying this as okay, it's a CD8 positive T cell because it has CD8 on the surface. I guess technically that's kind of tautologically true, but it's something that like I think if we were to take a look at that CD8 positive T cell versus another CD8 positive T cell versus a third one, obviously very, very different cell states. And I think as folks now with RNA-seq and other types of technologies to better understand kind of genome-wide expression
Hmm.
levels, and also things like
Hmm.
ATAC-seq to understand genome-wide chromatin accessibility states, can really start to understand the complexities of the different subpopulations that we're talking about here and the differences within them. So it's a little bit actually kind of that historical looking at things from a uh, single surface marker because that's what we're able to see. And therefore kind of presume that that's a unified class to now with the new technologies that are developed in labs like the Greenleaf Lab to kind of better understand those cell states, I think can really elucidate different areas of biology and ways to intervene in like kind of translational ways that are masked otherwise by that sort of like categorization just based on what the technology enables us to categorize them as.
Hmm.
But I think your question too was more on the actual intervention, not just classification, which deeply important part of that, obviously. And as excited as we are for kind of deeper biological understanding of cancer and supporting that work within academia and within the early stage biotech companies we invest in, I think what we got really excited about is the potential to intervene, right, and to
Hmm.
try to develop new ways of treating patients. And that can be just from a deeper understanding of the disease itself and therefore better classification of the patients for a particular therapeutic intervention. But also some of these areas I think where we're understanding nuances around the actual disease itself or the cell type itself, there are now novel technologies for being able to go and intervene in a very specific way, whether that be in the entire class of gene editing, base editing, epi editing, that whole world of targeting, but also other types of modalities too that really only the last number of years have we seen kind of a sufficient understanding and clinical de-risking, whether that be kind of small
Hmm.
molecules into heterobifunctional or kind of engineered glue spaces or other types of approaches on the biologic side. I mean, it does feel like I've had the fortune of entering this early stage therapeutic investment world at a time that there's been like a Cambrian explosion essentially of different
Sure.
therapeutic modalities that have only really scratched the surface of it. So that kind of engineering mindset, I think, fits really nicely into the new opportunities that are here regarding design of new drugs and the ability to intervene in these spaces that we now have a deeper biological understanding of.
Right. Very interesting. Do you think we should have a more bottom-up approach to how we view cancer in general? Meaning,
Yeah.
don't put too much weight on anatomy and really start to think more about the mechanism of disease and start from there. Obviously, there are complexities there. We need to have very accurate biomarkers to describe the disease. But do you think we should make more of an effort to be more bottom-up and more mechanistic in the way we understand and treat cancer?
I definitely think so, but I also think that that's the way that the field has naturally been evolving, right?
Right.
With, I mean, checkpoint inhibitors, obviously, and MSI high
Yeah.
patients is a good example of it, but more and more with these sophisticated understanding of the biology that enables the engineering of these more targeted therapies, and I use targeted kind of in a very broad sense here,
Mhm.
but um, that kind of necessitates thinking of the disease in ways that aren't just tissue of origin and kind of pathology based, right? Not to say that those aren't important as well.
Sure.
And I think that there's, will it always just be every type of cancer that has this surface marker is going to be amenable to this particular, ADC that targets that surface marker? Like, no, I don't think that's where we're going.
Right.
But as we have deeper understanding of the mechanistic pins that underlie the tumor itself. I think that that does help us think more creatively around, um, smart clinical development decisions that can bring those treatments to more patients that could benefit from it than just as well, it's this specific tissue of origin. That's the only patient
Right.
population we're going after.
Sure, the tissue of origin, I agree. It still has a lot of meaning and important implications. The way I think about it is it's a geolocation of where the malignancy is.
Yeah.
And the location itself matters because of the environmental factors, what we
Yeah.
call macro environment. Um, and one can hypothesize that uh, putting the environmental factors aside, uh, all tumors characterized mechanistically should behave the same. I'm actually involved in a very interesting uh, NIH effort called SysCan that is focused on manipulating the microenvironment in order to overcome primary and secondary resistance and tough to treat malignancies. It's very interesting. We've done a lot of work historically in that area. But for some reason, we still put a lot of weight into the traditional classification schema. So I'm so glad you mentioned pembrolizumab with MSI high. I have a very quick anecdote to tell you.
Awesome.
This the approval was in 2017. It was its first tissue agnostic indication when Merck submitted the application to the FDA. I was at the FDA at the time, as you know, and it was somewhat of a controversial decision that the FDA made, and there were even some folks at the FDA that had to really think it through to understand why uh, in that indication and in those specific patient populations, being MSI high and then ultimately tumor mutational burden high trumps histology and anatomy. So there was a learning that a lot of folks at the FDA had to go through. But I do remember a meeting we had when we before we approved pembrolizumab for MSI patients in a tissue agnostic matter with EMA. And it was tough to convince them, and they thought we were
Mm.
being a bit radical. And I remember Rick Pazder told the story. He's talked about it publicly, Rick being the head of the Oncology Center of Excellence. He basically told the story of why we have an anatomical classification, that it's not really science based. It's because surgeons used to treat
Mm.
patients with cancer and they had an anatomical approach. They saw a lesion in the thorax in an organ we call lung. Well, that's lung cancer. So there was basically it was just a matter of it's a historical artifact versus a scientific reality. But going back to then your ability and interest to understand complex systems, and in this case, the underpinnings of the genomic drivers of malignancies. And really, so at Stanford, you were exposed to the cutting edge of that. Um, but then you made a transition to one of the big three, Bain and Company. How did that happen? Was that also driven by your interest in understanding complex systems, but from another end, maybe organizational or financial?
I wish. I think that's too eloquent and generous, honestly. The reality of the situation is, as I mentioned earlier, I'd only ever worked really in early stage kind of tool development labs, and whether that be at BU or Stanford, or, I wanted to experience something different. I think, and was still evaluating: do I go down the path of kind of going to med school, doing a PhD, doing investment banking and getting the finance side through that, and consulting was just one of those options, honestly. So had talked with a number of different groups and ended up being an intern at Bain for summer, largely to just try something different. And
Mhm.
I think it was a really awesome experience. I joined out of undergrad as well, was there for a handful of years, mostly working in their private equity and biotech practices.
Mhm.
I learned a lot through that, and still, highly recommend that as a route for folks who are, wrapping up their PhD or postdoc or their undergrad and interested in kind of the business side of biotech, but not sure exactly what element of that they want to be in. I think it's a really awesome training ground for that. There's a lot I've learned from it. I think, in terms of, especially with their private equity group, what like
Mhm.
a high quality due diligence product looks like in that space. Really highly functioning teams working together efficiently. So a lot to kind of learn organizationally as well. But my one negative to it is do think if you're someone like myself who knew what I wanted to be doing, which is early stage science and the translation of early stage science, on the content side, sometimes it can feel, a little bit lighter. And I had the privilege of working with some amazing clients who were directly in the space that I, wanted to be in long term, who were launching incredible drugs. And
Mm.
it's a little bit different when you're working on kind of what's the right size for our sales force for this drug launch versus kind of, is this some great early stage breakthrough that needs funding to get through and turn into a new drug? And my interest just squarely lied in the latter. So while I thought it was an awesome training ground for a couple of years, I always knew I was going to be pivoting back into the early stage
Right.
world at some point.
Yeah. Seems like you wanted to be closer to the science
Exactly.
and translating that science to the clinic, which is uh, one of the most critical parts of the continuum, and where the risk is quite high. We can
Yeah.
get into your appetite for risk later when we talk about the investments that you're making today. But um, it would be great, Dan, if you could add some color about what made you choose Emerson Collective as your next sandbox after Bain. For those that maybe we can also explain to folks what Emerson is and how you transitioned there.
Yeah, absolutely. And look, I think Emerson Collective is a really unique organization, honestly, and one where I spent the majority of my career, and certainly the key part in terms of working with Reed to build out this oncology-related strategy. And frankly, it's really the only organization I'm aware of where we could have developed this exact strategy, because it fits really well into the overall objectives and structure of the organization. So it's a single family office based in Palo Alto, started by Lorraine Powell Jobs, and really has an interesting structure as an LLC, which maintains a lot of flexibility in terms of how to operate and what sorts of activities to do within the LLC. And I think takes an attitude of tool agnostic use of capital to have an impact in a couple of different issue areas that the organization cares about. And that can take a couple of different forms and a different mix and kind of different flavors of strategies within those financing types
Hmm.
within each of the different spaces. So, you can imagine how in education, there's going to be work on the for-profit side with ed tech companies. There's going to be work on the nonprofit side with philanthropy supporting nonprofits working in K through 12 education space. There's going to be work to do on the policy side as well. And, having an organization that can kind of plug in seamlessly across all those different components and develop a strategy around that, I think is, a really unique one to have an impact in a couple of these different verticals. So, Reed joined, the family office back in 2015 and started out the philanthropic part on the oncology side. And obviously, there's a lot of different ways that philanthropy can have impact in cancer, but what he really focused on was supporting project-specific academic research in cancer labs, let's say broadly defined. And from that, and those grants, by the way, are truly no strings attached.
Hmm.
There's no royalties or ROFR,
Right.
ROFIN, anything like that. It's a gift to the university to support work that's going on in a particular lab on a particular project. Um, and, while I was at Bain, I was talking with a number of early stage investment groups about working with them. And of course, had known Reed for a long time at that point. We had worked together in that lab together, but we're
Hmm.
also just friends. And, he was thinking about building out a for profit strategy within that cancer strategy of the organization, which again is not new for Emerson Collective. Emerson does a wide range of different investment activities, but
Sure.
was going to be new within the cancer space. So I viewed it as a really great opportunity to both get to do that early stage science oriented investment work that I was incredibly excited about, but also to do it in a little bit of a scrappier startup type environment where the two of us were going to work to build that strategy and the cohesion of the philanthropy and investment work. So that was why I joined. I think it's been an awesome decision. One of the big risks, of course, was working with Reed since we've been friends for such a long time. And
Right.
we worked briefly together in a lab, but that's a little bit different than something like this. And, pleased to say, obviously, that's worked incredibly well. And he's been an amazing business partner for the last decade or so. But, I think organizationally, what we both found was that philanthropic kind of academic support on the cancer side and the early stage venture investment work on the cancer side actually played together in a really seamless way, even more so than a lot of the other verticals that Emerson was engaged with. It's the exact same people and technologies that are involved on kind of both ends of that spectrum from the nonprofit capital to the for profit capital. So having an organization that had the flexibility to really plug in with both and then also help get from one to the other has was something that became immediately apparent as kind of a unique angle that we could help accelerate new science faster. So that was really the impetus for doubling down on the strategy, continuing to invest more philanthropically and invest more on the for-profit side and build out our capacity to help translate between the two.
Sure. That's amazing, and that's a line of work that you continue to advance at Yosemite, which we're going to get into. But when you were at Emerson, was there a strict divide between the grant portfolio and the venture portfolio? And for the grantees, was there any support outside of financial? And how did they approach you for maybe venture funding if the science was promising? And if you could educate us on that process, how to transition a grantee to potentially a startup.
Yeah, absolutely. And, everything that I say here about our time at Emerson, just to preview it a little bit, really is the same thing that we're doing here at Yosemite as well. And these elements of the strategy, we've maintained a lot of consistency around. One key component of that is essentially the purity of the grant work and the investment work. So we do keep it really separate in terms of not the team that's working on this, because we think that that translation between the two is a critical element of actually, making these things have impact faster and more efficiently. But there is a very strict divide around our grants are truly gifts and our investments are true investments. And There's a lot of potential shades of gray in between those two, and those are actually things that we try to stay largely away from. So on the philanthropic side, as I mentioned, this is something that really has no strings attached, right? We're not only no financial strings attached to it, but if we're supporting your work and it's this amazing breakthrough and you want to translate it out, but end up deciding to work with a different venture group to do it, you're totally empowered to do that as a PI. And wide range of reasons behind that tactically, but frankly, one of the most simple is we want people's best ideas and to be supporting their best, even if it's crazy idea through this philanthropic grant work. And we don't want them to be concerned around maybe they don't know us that well organizationally and they're wary of kind of signing up for a decades long engagement on the for profit side if things work out here. So maybe they've, worked on their last 5 companies with Third Rock and want to do a sixth with them. And like that's awesome. And that's still the type of science that we want to be supporting in academia if it's the right type of high impact work that could, have a great impact for cancer patients. That said, we also feel like it's on us to really earn our spot as someone that you want to work with to then translate your discovery if it does end up being something that's exciting and translational. So to your question around like what sort of support do we provide, it's of course there's the financial support of the project itself, but one of my favorite aspects of the job is going and meeting with professors, talking with that sort of work, and really trying to collaborate with them around what are the next steps here. Is there an additional grant? Is it a grant that comes from us? Is it a grant that comes from someone else in our kind of granting philanthropic network? Is this something that's ready for being spun out into a company? And if it is, are we the right partners to work with you on that? Is there someone else that we can introduce you to do that? So there's not one single formula. I think each individual technology and situation is its own unique situation. But providing that sort of thought partnership with folks as they are working on these projects, I think, is one of the good additional support elements. And I think, Sean, you've been to some of our cancer research summits in the past. So just hosting convenings like that where folks are able to talk about their work and collaborate together and potentially spur new ideas as well is another sort of support that we try to bring on the philanthropic side. I'll say in parallel to that philanthropic work being truly a nonprofit gift, though, on the for-profit side, we take a very similar purity to the approach, honestly. These are for-profit investments, and we're not trying to underwrite things that couldn't attract market rate capital to have an additional impact. We're making an investment because we think it's the best use of a dollar to get a great return back for our investors. And I think I'm super privileged that in the space of oncology, that tends to be extremely well aligned with patient impact. So it's not something that we need to struggle with very frequently. I think if you're going to have a massive clinical impact on a lot of patients with high unmet need, that's also going to be something that's kind of rewarded commercially too. But we do keep it quite separate. So, and I think that's important for a variety of reasons. But one of it is to scale up the operation and attract more capital in both areas. There are more people who, have an interest and ability in deploying for-profit capital for strategy like that for-profit strategy. And there are a lot of people who are interested in deploying purely philanthropic capital that's given as a gift. And then we can also work with them with the nonprofit strategy. And where we get the synergy, where we have the additional impact here is really in the synergy of the two. And that's different than other strategies for folks who are doing great work in impact investing, which, super broad term.
Right.
I think there's folks who view, additionality as a critical component of that, which in this circumstance would be like, we won't do a deal if MPM would have done that deal. That's not how we operate because we feel like our additional impact is between the robust philanthropic organization and for-profit organization and the ability to kind of, leverage both of those together to accelerate new cures faster.
And it seems like the grant portfolio also puts you guys in the trenches with the scientists. And
Yeah.
for me, it's been remarkable to see over the years the continuity in cases where the science has produced and has been successful, and then it's entered the commercial sphere. So that's quite a rare combination that a lot of other institutions, investors, and foundations don't have. So, having said that, how do you distinguish your grant making philosophy from more traditional sources of research funding? such as NIH, but also other foundations?
Yeah, look, I think that we try to have a really collaborative view toward this because I think that there's all sorts of different funding sources for academic cancer research, and really understanding that landscape so that we can best plug in where are we uniquely suited to fund this, where are we suited to fund this with a co-grantor, or where is this best suited for another granting organization that we're aware of here. I'll also just briefly touch on scale too, since I know that there's a lot of discussion around kind of federal potential retrenchment in different areas and things like that. Which we continue to be strong advocates for additional money into science and into cancer research in particular. That's a we're never going to touch the scale of the NIH or some of these other funding sources, so I do want to highlight that as a difference. But there are areas of research and work where I think that we are uniquely suited to be able to plug in and add catalytic capital in that philanthropic space.
Hmm.
We take a very broad view toward cancer and have supported work around in a couple hundred different labs globally, actually, over the last decade on range of different research topics. The ways that we currently actually operate grant wise, part of it is through our phase that we solicit, where we are sending out a request that in a particular topic area, whether it be our last year's ones in targeted toxins and cancer vaccines, or other types of topics that are within that sort of general scope, so broad enough that there's a lot of exciting research going on, but also specific enough that we can create kind of cohorts of grantees who are working on important problems in cancer that are related to each other and could potentially collaborate. So for those type of RFAs, we work with ACS as a good example on a really collaborative granting organization.
Right.
We both put up half the capital to support this work. We're both involved in the selection of the topics of the grants that come out of those topics and who ends up getting the awards. So whether it be them or many other granting organizations that we've done that kind of co-grant with in the past, I think it's really important for us in the philanthropic ecosystem to understand what the other players are here so that we can best work together and for appropriate types of projects be supporting those same projects.
Right. Yeah, makes sense. And it seems like philanthropic dollars can be quite helpful in supporting high risk, high reward ideas, de-risk them. Something that it seems like even the NIH is thinking about. I heard Jay Bhattacharya, new head of the NIH, talking about the fact that he wants to have more of a VC type approach to the NIH grant portfolio. And really optimizing for supporting high risk, reward research
Totally.
and capturing the asymmetric returns. Whereas today, NIH maximizes for success of every single program. Obviously, as you said, the scale of their investments is quite large. But do you think federal agencies like the NIH should be more entrepreneurial in how they support basic science research?
Yeah, absolutely. I think thinking really creatively around that is important, and I think that there is a gap that they can step into there. You know, I, the fact that a lot of that extremely high risk research goes unfunded is frankly why we've had such a gap to be able to fill
Hmm.
step into. But there are many more great ideas that we can't support, obviously. So more organizations, whether it be federal or other granting organizations, that I think are able to take that type of risk in the work that they're supporting philanthropically is really critical. I would say that in addition to that kind of high risk tolerance lens that we take, the other element that I think is maybe more VC like in terms of our granting is just this notion that we have a venture arm as well and can kind of overlay the translational lens, I
Mhm.
think, to some of this work here. And I think that's a double-edged sword, to be fair. I think that it's actually really important that we have organizations that are willing to take high risk, not just in translational areas, but really basic bio discoveries as
Mhm.
well. And that does require deep amounts of funding too, and thinking about it.
Right.
But I think that for an organization like us, being able to see, okay, this thing, if it works, is going to then be able to attract this sort of for profit capital to get it to that next inflection point, and then attract this sort of for profit capital to get it to that next stage, makes the grant writing a little bit more efficient in some areas where while we love supporting basic bio research too and things that are not necessarily as shovel ready in the for-profit world, what's a shame is if you support something that, ends up working out in like high risk, the science worked, which is amazing, but then ends up not being something that's really translational in any way, or that the insights are translatable to other areas, because then you supported a cool paper, but something that's
Right.
not actually going to get to patients. So
Right.
being able to filter that out as we think about what sort of
Sure.
support is
And
important.
it seems like you have a very unique ability to do that because you're involved both on the venture side and the grant side. And when you make a venture investment, you have to think about the practical realities of the science to reach the patient, the commercial aspects. So, does that translate into making grant decisions, meaning how practical is the science at the end of the day? Because there are a lot of cool ideas that people could have and from a purely scientific point of view and a mental exercise that could be interesting, but they may never translate commercially.
Yeah.
Is that something that uh, you do consciously, really thinking about the practical realities of the real world and then reflecting that in the grants that you make?
Yeah, exactly. And look, over the last decade or so, we've experimented with different types of grant strategies. I talked about how we do those RFAs, the ones that we currently do with ACS. That while we're involved in the selection of those topic
Mhm.
areas and are involved in the review process, we stand up independent scientific review boards to actually make the decisions around those. So on the flip side, there are other types of grants that we do where every investor on the team is involved in writing grants as well. And there's not a particular mandate on this does need to be directly translational with that and turn into a potential new co-incubation, but it is one where obviously folks on the for-profit side are thinking with that lens as they're collaborating with researchers. And importantly, I also think highlights, it's very easy for folks in the investment world to write stuff off as being, oh my God, that's just too early right now. Such cool science,
Yeah.
gonna, potentially have this huge impact, but it's too early for me to engage with the venture lens. And I think that having this other arm and the involvement of the investment team in that arm really takes away our ability to write off ideas in that same sort of way. Because, if it is really that amazing and potentially impactful, despite it being too risky at this stage for the fund to get involved with, you still have the obligation to engage with it and potentially
Mhm.
work to get it funded philanthropically. So I think that just changes the mindset of all of the investors on the team.
Sure. Yeah. You don't allow good ideas to fall through the cracks, basically.
And wait, one other thing too, in terms of difference on grant selection, I think the stage of people's career is really interesting around this. And if you look at our grantee portfolio, I'm super proud that we've got a really, really wide range of different sorts of professors that we're supporting here. And
Mhm.
it's everything from multiple Nobel Prize winners who started many successful startups to people who frequently this is actually their very first grant in a new lab. They were like a postdoc yesterday,
Mhm.
never gotten a grant before, but they might have some really exciting early discovery that has potential, but really high risk behind it. And frankly, while we're excited to support across the whole spectrum and work with a range of different people, those are always my favorite ones where it's someone who hasn't had that capital in yet to
Right.
explore these questions. And I think that we've seen some of the greatest discoveries actually within our grant portfolio coming from people of that type of phenotype. So excited to have a mechanism to help support those researchers too.
Yeah. Very interesting. And those are the researchers, the early career researchers that are impacted the most, it seems,
Yeah.
when it comes to the funding dynamics right now. Is that something that you've seen and been able to quantify? Are there more early career researchers coming to you?
We've definitely seen it anecdotally. Great question on quantification. We've done no work on that, but frankly, it's something that we should look into. I'd imagine that the data bears that out, though, for sure. Well,
Right, right. And you may make the argument that folks at that stage of their career, um, have the benefit of being naive, so they can come up with really audacious ideas that could be transformative.
totally.
So,
I mean, to just put in a plug for one example, right? Ryan Flynn, great grantee of ours, he's discovered RNA on the surface of cells. I mean, I'm sorry, that's like a fundamental, huge biology discovery. And back to the
right.
question on translation, like obviously many different directions to take that. But the stage that it's at right now, requires this sort of high risk philanthropic capital to really explore that further. And that's one where, I believe, first grant to his lab when he became a professor, and supporting that sort of kind of really basic bio, but a lot of translational potential in a lab that doesn't have sufficient funding around it, given the scale of discovery, is exactly the sort of work that we like to support at Yosemite.
Right. Right. Very interesting. Before we get to Yosemite, you got involved while you were at Emerson Collective and Tune Therapeutics as one of the co-founders, right? And you also stepped into the role of the CEO there. Can you tell us about Tune Therapeutics? The science is quite fascinating, and it seems like it draws from your scientific foundation, epigenetics.
Yeah, and I have to be a little careful here because I know that we don't have so much time left at the moment, and I can go on about Tune for forever. So if you'd ever
Right.
like to talk more about it, let me know, Sean. But it's look, I deeply passionate about the potential for epigenetic editing in a lot of different ways. But I guess briefly on the formation of Tune, it's a special company for us at Emerson and Yosemite, actually, just in that it's the first kind of new co-formation that we were really a critical driver in putting the pieces together for. And drawing back to that strategy around how we have the parallel philanthropic and for-profit work, it became pretty clear early on that we needed to build out capacity to engage at that company formation side. And that's not anything new in the private biotech financing ecosystem. Obviously, there's many different flavors of that
Mhm.
kind of company formation work that different funds take. But we realized that we had to have some sort of capacity and capability there to work with our researchers to help them turn these into investable assets that can accelerate their discoveries into patients. Um, Tune was our first one really doing that, but since then, of course, we've got many more examples of work coming out of the grantee portfolio and otherwise that we've helped get off the ground and spin out from academia. But what brought me to Tune originally was, in part, I think, just my interest, I'm sure, in chromatin structural biology and the potential for targeted expression manipulation, and some of the work that, frankly, we were seeing out of academia in
Mhm.
at risk of going on too much of a tangent here, actually in an area that is not related to the lead program of Tune, but in the epigenetic dysregulation of CAR T cells, but also other immune infiltrate in the tumor microenvironment for solid tumors. And, the understanding that a lot of that was epigenetic dysregulation, and the notion that we have these tools potentially to go in and rewire those cells to become more efficacious was something that got us excited about, taking a look at the space a little bit more deeply. And one of the nice things, as I said, about the flexibility of the organizational structure is we didn't really realize at the time, like, is this going to be something that requires more philanthropic capital to help de-risk things
Mhm.
in academia to show that we can push these T cells into a phenotype that is going to be better at killing the tumor cells as opposed to getting into the TME and then having some sort of exhausted phenotype? Or is it something that really is ready to go prime time right now and we can, get a company started and invest in it? So that was what originally kind of drew me to this space, is the potential for these epi editors, which, I'll briefly go into what they do, but that and the problem that some of our clinician scientists were seeing around uh, immune related therapies and solid
Mhm.
tumors. But so briefly on epi editing, this is a notion that takes advantage of really all of the work in the gene editing field over the last fairly long period of time here, and essentially just takes out the nuclease activity of whatever the DNA targeting domain is that you're working with. So whether that be CRISPR, Zinc or TAL, you need something to localize to a particular area of the genome. And then what we do that's different from all of the other gene editing approaches, like a base editing or an insertion or a knockout, we don't cut or nick the DNA in any sort of form. And instead, the localizing domain has fused onto it some other domains that affect the epigenetic state of the area,
Mhm.
whether that be like a DNMT to add, methylation marks to the area, or something to modify a histone acetylation and compact the chromatin. Some sort of epi modification domain that is now localized to a very particular region.
Right.
And the potential for this technology, as we realized pretty quickly, while we were originally excited about it in T cells and had filed IP in that space around it, and by the way, I'm still really excited about that and think that we've built out much more data, both the company and other researchers that have now shown that there is a lot of potential for that kind of phenotype tweaking in immuno-oncology. But we also realized there were a bunch of other very near-term translational applications for a technology like this, both within cancer and outside of cancer. And I think if we think about the promise of gene editing and the issues that the sector has faced, I think we've been really blown away by the amazing clinical results that folks have gotten in a range of different indications. But I think that we struggle with the high cost of the therapies and/or the relatively small market sizes of some
Right.
people's here, and the combination means kind of a lackluster commercial performance, which has then, I think, turned away investment interest in the space relative to what we think it should be. We think that something like epi editing has a potential with relatively low cogs and to really intervene in some massive markets with large patient unmet need here. And I think that that's really exemplified by the lead program of Tune Therapeutics, which is in the hepatitis B space.
Hmm.
And I think really fortunately too, that of course is core to our mission at Yosemite to make cancer non-lethal in our lifetime. Hepatitis B is the largest driver of liver cancer globally. The scale of this disease, frankly, is just mind-boggling. Every time we kind of build a model and take a relook at the numbers here, it's hard to wrap your mind around. We have probably around 2 million patients in the U.S. with chronic Hep B, although a number of them underdiagnosed. But globally, we're talking about a disease with over a 250 million patients. So the potential for patients broadly on a global level for kind of reduction in mortality from HCC, we think is really massive for a potential functional cure for hepatitis B. And it's a very simple drug. It's a LNP directed to the liver, where we work with the Acuitas on our LNP. And it's a mRNA that encodes one of these epi editors and a guide RNA that brings it to this regulatory region of the hepatitis B genome and can interact with both the integrated DNA and the triple C DNA to essentially methylate the site and compact the chromatin and then shut down the viral expression. We dosed our first patients last December while I was still operating with the company, and our dose escalation since then. So tragically, I can't talk about the data yet, but stay tuned
Right.
for that company. But I think more broadly, the potential for a therapy like this in hepatitis B is massive, but also for this as a technology outside of that too. You swap out that guide with some cardiometabolic guides to lower cholesterol, and I think it's something that has the potential there to, in a multiplexible way, I think unique to a lot of the gene editing approaches, be a one and done treatment for cholesterol reduction,
Right.
certainly for folks with familial hypercholesterolemia, but also potentially more broad populations that a low COGS one and done IV infusion that with durable responses has the potential to have real impact and commercial traction. So very excited about it more broadly. I can go on forever also on how we think that it's the right approach for walking a cell down a Waddington landscape as we think about the future of like iPSC-derived therapies. And back to something we talked about toward the beginning, the actual more comprehensive expression states of those cells we think is really critical in terms of clinical efficacy of the products that you're creating, not just the surface markers of those cells, and the ability to actually, in a more sophisticated, targeted way, up and down regulate these genes during the differentiation process can build a more efficacious cell type for treatment of patients. So anyway, I'll pause there, but like wide range of different applications, I think, for epi editing as a modality that, we're incredibly excited about. Hep B is really the first iteration of this for the company and for the field, but think more broadly, there's a lot of opportunity here for epi editing.
Right. Well, it was exciting. Looking forward to the clinical data when it's publicly reported.
Yeah.
Dan, do you think that what you said earlier, that in cancer research and cancer care, usually success depends on impact, patient improving the patient's life, survival, efficacy and safety, essentially.
Yeah.
Do you think that equation falls apart if we're talking about therapies that have curative intent?
Yeah, it's a great question because, and I've been frustrated too, in part, definitely from the lens of being an operator at Tune has helped shape my frustration with elements of this. Where I was just talking about the potential for these one and done, cholesterol reduction drugs, and I think, outside of oncology, the equation does become a little bit trickier and guided by a lot of realities around the kind of, frankly, the payer ecosystem here domestically in the US with respect to one and done treatments. And I don't know how this will evolve going forward, to be honest. I think that if you're to talk to patients or providers, just to use this kind of cholesterol example, folks are pretty excited about a one and done IV infusion with no gene editing that has that sort of dramatic reduction in your LDL-C. And that's a space obviously that from a modality perspective is quite crowded and has a lot of competition. And I think that ranges from everything from Repatha to statins, and obviously Verve is a very clear example here on the base editing side too, for more of the gene editing element of being able to intervene. And so whether there's commercial space, I think is totally debatable. We think that the
Mhm.
durability of this, the multiplexability, et cetera, is really exciting. But I think that calculus is a little bit different if you are thinking about this from a comprehensive healthcare economics standpoint of, for example, a single payer system country where, is it beneficial overall for a relatively cheap
Right.
one infusion versus keeping everyone on statins and including the low compliance rates? Like that is, I think, a very clear calculation. That calculus just doesn't really fully work here. And I think that filters through to investors and pharma companies as well as they think about the potential for these products. So how that evolves over time regarding the right type of payment structures for things as we're getting more and more clinical evidence of the potential for one and done treatments in some chronic diseases. I think it'll be really interesting to see how that evolves. But what we're hopeful for is, I think it's the right thing for patients and the right thing overall for healthcare economics.
Mhm.
So I'm optimistic that as a society we're going to find out a way to get those into.
Right. Well, let's hope so, Dan, because I think our healthcare system, is uniquely fragmented in the
Yeah,
U.S. And how we deliver care, how we prioritize certain research themes is all related to payment,
yeah,
unfortunately. And that ecosystem, it seems like, has remained uh, quite stagnant, where our science has really progressed exponentially.
yeah.
So I think it seems like we have an infrastructure problem, that our infrastructure was built for a world that no longer exists. And I'd like to be optimistic, Dan, and say that organically it'll evolve and accommodate these potentially life saving curative therapies. But I think we need both top down and bottom up pressure.
Yeah,
A topic that maybe we can talk about at a later time.
makes sense.
Uh, so I know we're almost out of time. Time goes by fast when you're having fun. But I'd like to better understand the transition from Emerson Collective to Yosemite. Why Yosemite? It seems like Emerson Collective is such an amazing and unique sandbox. What was the impetus behind starting Yosemite?
Yeah, and I'll start out by saying you're totally right on Emerson being a unique, amazing sandbox. And as I mentioned earlier on, like this is a strategy that could really have only been built within that ecosystem. It's an organization that gave out large sums philanthropically to support this cancer research in no strings attached ways. And it's really tough to do in a for profit world if we were to start there by saying, hey, we have a fund first, and then we're going to try to do philanthropy. I don't think that would have worked. You needed an investor and donor who was really willing to build out those both in parallel and see the potential for impact because both were built out in parallel. And then over that course of since 2015 through today that we've really built out that work at Emerson Collective, I think that that's kind of been the proof point for the model. We can point to this really working, to de-risking a lot of awesome science in academia, for that directly turning into companies that we're able to invest in and that are already having massive impacts for patients and returns for investors. So that sort of evidence that we were able to build within the family office, I think, equipped us well to spin out the strategy. And that's not because of any limitations within Emerson Collective. We just thought that, it's something where we can really benefit from bringing more investors and donors around the table to help support both of these strategies. And it's not something that we're unique to as well. Emerson's been an incubator for a number of things that then have kind of
Mhm.
spun out and attracted other outside capital. But in this case, been super pleased with basically being able to operate the exact same strategy that we were at Emerson with the same team, but now just have a more diverse set of investors. And that ranges from, academic endowments and hospital systems who are really mission aligned with the type of work that we're doing, but also care about returns for their endowments, obviously, to other family offices that both care about return on their investment, but also likely care about, cancer and the impact in cancer. And some of those are also direct donors to our donor advised fund, which is a vehicle that we do our
Mhm.
from
Mhm.
as well. So it's been a really seamless transition. We're keep doing the same exact work that we were doing before. We're involved with the exact same companies that we were before, and I think evolving that strategy as the ecosystem evolves, but being able to kind of now work with new partners on the LP side who are able to support this work too.
Sure. That's fantastic. So then, what signals do you look for to decide if an idea that's been incubated in the research setting is ready to be spun out?
Yeah, it's tricky, right? Because I think that this is quite different depending on the exact space and what it is that we're really looking for here. I'd say we take more of uh, an area specific approach to really understanding what is it that we're going to want to see in that space to have confidence that this is ready for a profit dollar instead of a philanthropic dollar. That ranges pretty significantly. I mean, we've been just as one small example on this, we've been supporting a lot of work philanthropically historically in the microbiome space. I think that the correlative work around checkpoint inhibitor response and microbiome populations is extremely robust data now over the last number of years that's been developed by a bunch of folks, and the mechanistic insights in terms of what's actually driving those differential responses has been what we've also been supporting work philanthropically exploring to try to get real confidence in. And in that specific kind of line of therapeutic thinking, what we're really looking for here is that understanding mechanistically on what's driving that to then think about translation into a therapeutic and that path of that therapeutic development.
Hmm.
So that line for us is like once we get confidence and this is the thing that's actually changing responses, and there's a clear way to intervene with this sort of therapeutic, and there's a clear clinical developable path for that as well in this setting. That's what we're looking for that particular subset, and that's going to be
Right.
different from that to other types of modalities, indications, etc. But for each one of those, I think having a clear perspective on what is the data that's going to move us from nonprofit to for profit.
Right. Makes sense. Seems like the prerequisite is embedding yourself in the science,
Yeah,
uh,
absolutely.
which is quite a complex undertaking, but it seems like Yosemite has been able to accommodate that quite well. So Dan, one last question, and so you're exposed to a lot of amazing science, usually at the early stage. What are some of the things that you're seeing that we may not be familiar with yet that you think will make a difference in the next 5 to 10 years?
Oh, great question. I mean, can go on for far too long around this. Some stuff just look. I think that we're two broad categories, right? One in terms of what I was talking about earlier on this Cambrian explosion of different highly engineerable modalities, right? And this can range from ADCs, radiopharma to autologous cell therapy
Mhm.
pills, and I think that for each of those, there's a little bit of a tip of the iceberg kind of moment. And where we've seen this work clinically and in some cases commercially as well, but there's so much more room for improvement, whether that be on the ADC side, different types of targets and patient populations, different payloads, to something like in the neoantigen vaccine or TIL space, where I think we're seeing exciting early results and kind of improvements around manufacturing and ability to turn this into really scalable products for more patients is really critical. So kind of engagement in those areas which have a proof point but have a lot more potential here, and trying to pull the levers on how do we bring that to more folks is one broad category that we focus on. And then I'd say the other one that I'm excited about are the really early new stuff still too. Like, and
Mhm.
one example on that, I'm a firm believer that we're only scratching the surface of induced proximity and what that can really do. I mean, the development of the PROTACs, of course, in the first real engineered approach, and then the retrospective looking at thalidomide to understand the mechanism there. Like, these are things that are really doing stuff in cells and in actual therapies. And I think that the potential to bring the ability of bringing two things close together is like
Right.
well beyond just degradation. And even just within degradation, there's so much more we can do too. So as one example of, I think there's very early, very cool work that as we go down kind of 5-10 years, we're going to see a lot more things coming out of that world.
Right. It seems like the life sciences is getting closer to the physical sciences.
Yeah.
Uh,
And
bring a
epi
lot
editing,
more.
another plug for that one too. But epi
Right.
editing is going to have a big impact.
Absolutely. Dan, it's remarkable that we've talked for an hour and we didn't mention the word AI once.
It is actually. Yeah, totally.
So maybe next time. Well,
Yeah.
thank you for your time. This was a fantastic conversation.
Awesome. Yeah, thanks so much, Sean. Really great to meet you.